So welcome to the Department of Physiology seminar. Today, we are really excited to welcome Dr. Rachel Green from Johns Hopkins to give the seminar today. Rachel received his bachelor's degree from University of Michigan in 1986. And then she went to Harvard and worked with the Nobel laureates Jack Szostak and working on ribosome and self-splice introns. And then during her postdoc, she worked with another Nobel laureate Harry Lauder, and of course was known for solving the ribosome structure. In Harry's lab, Rachel focused on understanding the ribosome functions and the interaction with RNA and how it regulates translation. In 1998, she joined the faculty of Johns Hopkins and stayed amazingly after 20-some years. And she's now the Bloomberg Distinguished Professor of Johns Hopkins in the Department of Molecular Biology and Genetics. Some of you probably know our chair, DJ, used to be there for some time. She's also a HHMI investigator. Rachel's lab has focused on translation, regulation of translation, ribosome functions, and how protein and mRNA quality control pathways are regulated during his time at Johns Hopkins. I personally regard Rachel's lab as probably the most influential lab in the world in the field of translation, specifically on translation regulations. What impresses me the most is the ability of a lab to combine the state-of-art technology to really address fundamental questions in the field. And her discoveries are just too numerous, so I won't mention too. So needless to say, she made really numerous seminal contributions in the field of translation regulation and quality control pathways, discovered many of the key mechanisms and components. And because of her contributions, she was elected to the member of the National Academy of Sciences and also a member of the National Academy of Medicine, and also a member of the American Academy of Arts and Sciences. So that's quite a lot of titles. So the title of her talk today is "The Collision of Ribosomes Are Critical Activators of Cell Signaling, Psychosignaling Pathways." So welcome to Dallas. Okay, it's your turn.
Perfect. Well, thank you for the really nice and generous introduction, Yi. That was really, really nice to hear. I should add to all of you, I'm happy to speak to all my colleagues in Dallas. I'm actually in DJ's previous office. And so this is where he used to directly live. And I had a nice talk this morning with Josh Mandel, who was a colleague here as well, and Nick Conrad, who was a graduate student when I first arrived at Hopkins. So it's really nice to see everybody. I'm getting faces on my screen here appropriately. Okay, so what I thought I'd do today is tell you a related set of stories from the past few years in my lab that all originate from our sort of deep interests in ribosome structure and function. And as you mentioned, I got my early training in Harry Noller's lab, where we really cared about the details of the inside of the ribosome and how mechanisms worked. But as time has gone on, we've increasingly sort of looked beyond the walls of the ribosome to ask how they play other roles in the cell. So, my new introduction to my talk starts with this slide, which is actually a cryo-electron tomography image of yeast cells from Liam Holt's lab at NYU. And what you can see here in gray are organelles that are highlighted. In yellow are actually some nanoparticles that he put into cells to measure viscosity. And shown in false colored in cyan here are ribosomes. And in this study, they were able to measure the concentration directly of ribosomes in the cell at about 23 micromolar and about 20% of the volume of the cell. And in fact, they found that if they played with the TOR pathway in yeast to change the concentration of ribosomes, the viscosity in the cell changed. So suggesting that ribosomes really are a very dominant component of the cytoplasm. And so the story I'm going to tell you, which is that we think ribosomes can function directly as signaling pathways to sort of as a readout for cellular distress, that idea shouldn't seem too surprising. So to get started, I'll remind you all that in bacteria and eukaryotes, where all the work today I'll be talking about, ribosomes move along a messenger RNA in a 5' to 3' direction. In eukaryotes, the messenger RNAs are capped and polyadenylated, and those are the features that get them out of the nucleus. What we think is that ribosomes load at the 5' cap and they scan until they hit the start codon, and they elongate through the open reading frame until they reach the stop codon and they dissociate. And we think we understand something about translation. What we think is that ribosomes are carefully loaded on at an appropriate speed, such that on average, ribosomes are nicely spaced along the messenger RNA, much like cars on the road used to be when we went to work in the morning. And avoiding, and what we know is that flow is most efficient when there are no collisions or slowdowns that result from objects getting too close to one another. So that's certainly been our traditional view of how ribosome loading and translation happens in everyday cells. For a number of years, we've been interested in the following process. We've been interested in what happens in cells when there's a problem with the messenger RNA. And that problem could be encoded or it could be environmental, like a damage. And what we've known is that when ribosomes encounter problems in the messenger RNA, there's a whole collection of quality control pathways that are triggered. And what we think is probably most important is you can imagine that if a ribosome doesn't reach the stop codon, the nascent peptide is incomplete, and it can aggregate and be toxic. On average, it probably is. And so we think probably the most important function of these pathways, these mRNA surveillance pathways, is to target the nascent peptide for degradation. We know in yeast that mRNA degradation is tightly coupled to these quality control steps when you recognize that there's a problem. And we know that ribosomes need to get off because in the absence of reaching the stop codon, they need a new mechanism for being released. And we've studied these pathways for a number of years. And the car analogy is that this is much like a car with a flat tire or something. You have to pull it off the side of the road and deal with the problem before you put it back on the road. So for a number of years, we wondered how it was that the cell would discern that this lead ribosome was somehow stuck and going more slowly in order to target this entire, to activate this entire set of pathways. And the most important insights into this came a number of years ago from actually Hanny Zayer's lab at Washington University. He was my former postdoc. And what he found through a beautiful set of biochemical experiments was that, in fact, it's not a slow ribosome that signals a problem, but that a collided ribosome signals a problem. And what happens is that collided ribosome provides a new interface that didn't exist before to recruit an E3 ligase that ubiquitinates small subunit proteins in order to recruit various factors that bring about this whole collection of outcomes. And so it's not just that a ribosome is slow, but it's slow enough that delay leads to collisions to happening and then to the activation of these pathways. And we've studied these pathways for a number of years. We've identified nucleases involved in them that target the messages for decay, factors that recruit nucleases, and so on. But what I'm going to tell you about today is that we think that these same collisions that can activate these mRNA-specific quality control events can in fact activate general stress responses. And you can imagine that might be required when there are so many ribosomes that are stuck on so many messages, for example, with environmental damage from UV, that in fact, there's a huge problem, and that the answer to the huge problem is not an mRNA-specific quality control pathway, but a general stress response. So that's where I'm going. I just wanted to give you a view of the collided ribosome. After Hanny's beautiful biochemical work, cryo-EM structural biology came in and really gave us a great look at the problem. What you can see here is the lead ribosome of a collision, the lagging ribosome of collision. At the interface, the two small subunits of the ribosome come together. You can see at the interface of these two small ribosomes coming together, some of the key proteins that are ubiquitinated by that key E3 ligase, US10 and ES10. RAC1 is a key ribosomal protein that meets at the interface. And in fact, RAC1 comes up in every genetic screen for these quality control pathways. And importantly, you can see the messenger RNA extending from the lead to the lagging ribosome. And you can imagine a space-filling model. It's actually not accessible to nucleases. And that's going to be an important biochemical handle on isolating colliding ribosomes. So that's just a quick look. how did we get to thinking about this problem? I'm going to tell you a quick story about earlier work in yeast to tell you how we got there. And the tools that I'm going to describe here will come into play in each part of the talk, so it's worth paying attention. For a long time, we've been interested in using ribosome profiling to get at ribosome function in living cells. And you're all aware, this came out of Jonathan Weissman's lab in 2009. You're all aware that basically the method involves cracking open cells where ribosomes are displayed all along the messenger RNAs at their sites of action. And the goal is to sort of stop translation as quickly as possible in your population of interest. Then to use a nonspecific RNase digestion, ribosomes protect a small fragment. You clone and isolate that small fragment and align it to the transcriptome. And from that, you can get information on where all the ribosomes in the cell are sitting in condition 1 versus condition 2. A key step in this process is how to stop translation. The earliest studies used a drug called cyclohexamide to stop elongation, and libraries that were made from cyclohexamide-treated extracts resulted in a very coherent set of ribosome footprints, about 28 nucleotides in length, which makes a lot of sense. And when you align these to the transcriptome, you get results like this. This is an average gene or metagene analysis of all reads aligned to start or at stop. And you see features here that look recognizable, where there are few ribosomes in the 5' UTR, a nice peak of ribosomes at start, 3 nucleotide periodicity throughout the open reading frame. Same thing is true at stop, nice peak at the stop codon, and no ribosomes in the 3' UTR. So there are a lot of things that look perfect in these data. But what was clear in these earliest studies was that certain things that we anticipated would be true in these data were not. We couldn't see them. We couldn't see that slow codons were slow and fast codons were fast. And when we starved cells, we couldn't see pauses where we expected them. And it turns out this method of using cyclohexamide blocks the elongation cycle at a certain point. You'll recall this is how ribosomes elongate. They have a peptidyl tRNA. They let in a tRNA for decoding. They make a peptide bond, and then the whole thing moves. This is the cycle of elongation. What happened, we think, in cyclohexamide is ribosomes all accumulated over here. And so the positional information that was here, for example, was lost through movement in the lysate that only trapped this step. We worked on methods to trap the ribosomes at 2 different steps using a different antibiotic, tigecycline. These methods allowed us to generate libraries that actually had 2 distinct footprint sizes, one at 28 nucleotides or 29, and the other 21 nucleotides in length. And we were able to assign these 2 different sizes to different states of elongating ribosomes. At the top are the 21 nucleotide reads, and these 2 other states correspond to the 28 nucleotide reads. So for us interested in mechanism, this was a powerful tool to look genome-wide at the states of elongating ribosomes. So with this tool in hand, we were interested in sort of asking, are there global changes in ribosome state as a function of stress? Actually, I forgot to say something. So how do we test whether this is working? The way we tested whether this was working was we recognized that 21 nucleotide reads should represent starving ribosomes. So we simply asked, if we starved for histidine, did we have an increase in occupancy at the histidine codons? And we did that experiment, and you can see the results here. What we have is untreated cells. We looked at 21 nucleotide long reads and histidine starvation, either in wild type or histidine starvation. There's 64 codons, and this is a pause score. And you can see using the old method that there's a little bit of pausing at histidines, but it isn't really impressive. By contrast, our new method using tigecycline as well as cyclohexamide yielded substantial pausing on histidine and convinced us our method was working. So we were really excited to have this method in hand, and we wanted to ask a biological question. So what we did is we took yeast cells, we exposed them to various stresses. We simply asked first whether the distribution of read lengths changed, 21 versus 28. You can see when we exposed cells to stationary phase stress, there was no effect on the distribution footprints, suggesting there wasn't much of interest for us going on there. However, when we exposed them to oxidative stress, we saw something quite striking. We saw an accumulation of 28 nucleotide long reads and a substantial depletion of the 21 mers. This is cell-wide. And we recognized that that meant that in this elongation cycle that I drew for you, the ribosomes were getting stuck here. So it turns out, and that's of course across the cell, turns out we weren't the first to discover this. And this result simply highlighted an old result, which is that it was known that oxidative stress in yeast activated the HOG1 MAP kinase signaling pathway, which was upstream of EEF2 phosphorylation. There's an EEF2 kinase here. And EEF2 is the key factor that's involved in promoting the movement from this state to this state. And that overall blocks protein synthesis. And so what we had revealed in this profiling experiment was a cellular stress and sort of the activation of an entire signaling pathway that led to sort of a stress response that blocks ribosomes at this step. So that was fun. Another analysis we did with those data was we said, okay, let's look at the 21 mer reads, which are these, and see if they look the same in wild type conditions or oxidatively stressed conditions. Those data are shown here. Again, there are 64 dots here, wild type cells, oxidatively stressed cells. And here we saw something odd. We saw that there were a couple of codons where ribosomes are disproportionately found in oxidative stress. Turns out these are all proline codons, and there's something to study, and we've only done a little bit, but it suggests there's an issue with proline metabolism. We did another analysis to look at proline pausing, and we did another metagene analysis. And so what we've done here is a metagene analysis aligning reads at proline. And you can see in unstressed cells, proline's a little bit of a slow codon already. And in oxidatively stressed cells, it's really a slow codon, genome-wide. And in fact, it's such a slow codon that we actually see a little peak back here, 10 amino acids back, which is actually represents a colliding ribosome. We know from that structure that I showed you, it's 10 amino acids from A site to A site of the two ribosomes. And that's really what got us started thinking. We knew that oxidative stress was leading to the activation of MAP kinase signaling pathways in yeast. And we saw that there was a collision here, and we began to wonder whether these collisions that activate mRNA-specific quality control pathways might also activate general stress response pathways in response to these genome-wide problems. And that really was the beginning of the stories I'm going to tell you today. I'm going to tell you two stories that are somewhat related and bring them together at the end. The first is a story of a MAP3K ZAC that we think it directly recognizes colliding ribosomes, and of EDF1, which was previously known as a transcriptional coactivator, but which also recognizes colliding ribosomes. And we think they work together to promote output, cell signaling output. So the first is the story of an outstanding postdoc in the lab, Colin Wu, who's now at the NCI. And that was all of his work in yeast. And so he knew that in yeast, oxidative stress activated P38 in signaling pathways in yeast. And it turns out that's true in mammals as well. Oxidative stress triggers this output. We were interested in understanding how this stress might trigger this output. We thought we saw colliding ribosomes, but we really wanted to know whether that was the proximal signal. And in having a conversation with my colleague downstairs, Sergi Rougeau, who studies MAP kinases, he mentioned to us that he studies P38 signaling, and his favorite triggers for these pathways, they're fast and potent, are three drugs, anisomycin, cyclohexamide, and ricin, which, as I alluded to before, are actually three different drugs that target elongating ribosomes. So that sounded interesting. It sounded to us like three drugs that target the ribosome are activating signaling pathways. And in fact, Bruce Magan's work through the 90s and 2000s argued, he called this the ribotoxic stress response because he recognized that it was ribosomes that must be activating these kinase signaling pathways. So we were interested in exploring this and exploring the possibility that maybe it wasn't single ribosomes, but colliding ribosomes. So, and I have to add here. I'm going to add that Bruce Magan also showed that UV irradiation activated this so-called ribotoxic stress response, and I'll get back to that at the end. So we started by just simply seeing if we could recapitulate Bruce Magan's results by taking anisomycin and seeing if in the presence of anisomycin, P38 was activated. It is. We then asked the question if it was elongating ribosomes, actively moving, or just empty ribosomes that were relevant. And we did that by adding a drug called harringtonin that binds to initiating ribosomes, allows all the other ribosomes to run off the end. And we then asked if that blocked activation by anisomycin, because we pre-treated with a harringtonin. And you can see, in fact, that the activation is blocked, consistent with the idea that it's moving ribosomes that are the critical signal for activation. We then wanted to ask the critical question, which is, okay, fine. Is it true, like it was true for mRNA quality control, that it isn't a stuck ribosome that's the signal, but maybe instead it's a colliding ribosome that signals a problem? So to do this, ask this question, we used the trick that Hanny had used in his paper, where he took cells and they were either untreated, where there shouldn't be very many collisions at all. He treated them with a high drug dose, in this case anisomycin, where we anticipated that all the ribosomes would bind to get stuck, or an intermediate dose of drug where the occasional ribosome might bind to the drug and the lagging ribosome will collide. And we reasoned there would be more disomes in this intermediate treatment. To look at this, we used that trick I mentioned, which is we used RNase to fully digest all the free RNA around the ribosomes. We separated these lysates then onto sucrose gradients. And you can see the predominant species is an ADS ribosome with a small amount of resistant disomes. We then did the same treatment for the intermediate drug treatment and you can see now the appearance of an increase in size of the disome peak, which correlates with the fact that this treatment leads to an increased number of collisions in the cell. So with that tool in hand, we were able to do what we wanted, which was to ask, is it at intermediate or high doses of drug that these pathways are activated? And we first looked at both P38, the homolog of yeast, and junk, which is another P38 kinase. What you can see here is if you look at activation of P38 or junk with increasing amounts of anisomycin, is that these pathways are activated, at intermediate, but not high doses of drug. So that was exciting to us and suggested that in fact collisions might be at the heart of this activation. We then went on to look at the activation of eIF2 alpha phosphorylation because work in the field had been coming out and we wondered whether eIF2 alpha phosphorylation by GCN2 in fact would be also looking at colliding ribosomes, and we did the same sort of experiment. We looked at its activation, and you can see it too is optimally activated at intermediate but not high doses of drug. So that was exciting, and it suggested to us that in fact, maybe downstream of many stresses, for example, oxidative stress or anisomycin, are colliding ribosomes, and that's the key signal for activation of MAP kinase signaling pathways with an output at the end of P38 and junk activation. You all are aware, however, that upstream of P38s are MAP1Ks, are MAP2Ks, and MAP3Ks. And I won't show you the data, but we actually did a CRISPR screen to look for sort of resistance and sensitivity to anisomycin. And in that screen, we identified ZAC, which is a MAP3K. It's called MAP3K20. It had actually previously been implicated in these pathways by Bruce Magan, but we found it again in our screen, and so we were excited about that. We did a simple experiment where we made mouse or ZAC knockout cells, which are shown here. You can see that this is the activation we see of P38 in the presence of anisomycin in wild type cells, and that activation is blocked in ZAC knockout cells, consistent with the idea that ZAC is at the top of this pathway. In data that I won't show you, we show that ZAC was also critical for activation of the integrated stress response, and that the GCN2 kinase that was involved in that activation is in fact GCN2 itself, which has since been shown, or its partners, GCN1 and 20, have been shown to recognize colliding ribosomes. There's a beautiful cryo-EM structure from Daniel Wilson's lab. So this is looking like ZAC kind of is the hub that sits at the ribosome possibly to activate these different signaling pathways. We next asked a simple question, which is, as you are all familiar with, I'm sure, at the ZAC or MAP kinase signaling pathways always involve phosphorylation. So we asked whether ZAC itself becomes phosphorylated as a consequence of collisions. What's shown here is what you've already seen, which is that intermediate doses of drug, P38 is turned on. What we did next, however, was to look at ZAC phosphorylation. To do this, we used a phos-tag gel because we didn't have a phosphospecific antibody, and this is a beautiful phos-tag gel where ZAC is completely unphosphorylated at low doses and high doses of drug, and is extensively phosphorylated at intermediate doses of drug, allowing us to argue that ZAC is activated as a consequence possibly of collisions, and that leads to then downstream activation of these pathways. Another obvious question we wanted to ask was, okay, that's great, but is ZAC really directly recognizing ribosomes? Is that the biochemistry that's involved? And I'll just show you two little experiments. The first thing we did is simply fractionate lysates, either untreated, low-dose drug-treated or high-dose drug-treated lysates. We fractionated them across sucrose gradients, and we asked whether ZAC migrates with elongating ribosomes or with collided ribosomes specifically. We were excited to see that in fact ZAC is clearly a factor that is migrating with the polysomes. I can tell you many people talk about proteins that associate with elongating ribosomes, very few associate this well. These are really, this suggested this factor really was surveying ribosomes, but it clearly wasn't only out there under conditions where collisions were occurring. So we took this experiment one step further, and we did that RNase digestion that I've shown you before, and asked where ZAC is activated in these lysates. And what I'm showing you here is an RNase digested lysate, fractionated across the sucrose gradient. This is the monosome peak, fraction 4. This is the disome peak in fraction 6. And what you can see if you follow ZAC activation is that the activation of ZAC is predominantly happening in this peak here, which is the disome peak. In fact, we think phosphorylate ZAC sometimes falls off after it's phosphorylated, and we can talk more about that, but that the unphosphorylated species are less associated with the disome. So all of this we thought was good evidence that ZAC associated with collided ribosomes. We have more biochemistry to identify a domain of ZAC that's responsible for this, and there's more biochemistry to talk about, and you can ask questions about that if you're interested.
So Rachel, can I interrupt for a second?
Yes. Yes.
So, so one question is this activation is concentration dependent of the drug you use.
Yes.
At intermediate concentration, you have peak activation, but at high, you lost the activation altogether.
That's right.
If I looked at the particle profiling you showed over there, I mean, the ZAC appears to be associated at both high and low concentrations. Right. So the association itself, maybe there was something. What is the high concentration of the NS2 to block this altogether? Does the disome disappear, or collision completely disappear, or is it?
Well, what our biochemistry suggests is that at high doses of drugs, there's no more collisions than in untreated samples, because what you kind of just hit them all at once and they all stop, and that's what that sort of earlier slide showed. We think the low dose is what gives us an increase in the actual disome peak, but what these data suggest, as you point out, is that ZAC, it's binding in at least, what we think it is doing is surveying. We think it's already bound to ribosomes, and it's surveying sort of the elongating polysome, the elongating polysomes. But it's only on, collisions happening that it gets activated. So it's always there, but the activation is specific to collisions. That's
so what I don't quite get is, at high concentration, why does the disome disappear?
Because it doesn't disappear, it doesn't appear. We think that it doesn't appear because we think all the ribosomes bound the drug and they all stopped. And so there was no collisions because none of them are running. So
for a given mRNA, where does this disome occur? Anywhere? How frequent is this? I've seen several papers sequencing disomes, but
yeah. So under normal conditions, it's slow sequences that tend to accumulate some disomes. So slow sequences are things like polyprolines. Sometimes there's some peptide sequences that get stuck in the tunnel, but in these conditions, there probably is no specificity because it's just any ribosome that's elongating. So if we did a disome sequencing here, it wouldn't be interesting because it would be everywhere. But we could get back to that.
Okay. So I just want to remind all the audience that if you have questions, either you raise your hand or you type your question in the chat box. So,
yep.
Okay.
Okay, I'll start again.
I think has his hand raised.
DJ, yeah. Should I go on or should I wait? Hello? Can you hear me?
Yeah, now I can speak. Yeah.
Yeah.
If you could go back, Rachel, could you go back to the polysome profiling and going to the.
Which one? This one?
Yeah, this one. Exactly. So I'm looking at the peak, right? So at the disome, so you see more disome at the low ANS. That's the idea.
Well, I'm just going to go way back here because this I think will be more effective. Right. This is the key one here. Okay, this is the idea behind this treatment is that untreated cells, none of them are colliding, more or less. High drug, more or less none. And that's demonstrated here because these are now RNase digested, but at the intermediate drug dose, you get more disomes.
Yeah.
So in this one that I'm showing you, it's a little confusing if you're not a ribosome person. This one, these are not digested.
Okay.
So there's no digestion here. And so this is just all of the elongating ones. But the first observation is made here, even though we know at low drug treatment, there are more disomes, and they'd be everywhere, right? Because they'd be in the middle of a message, they'd be at the end of a message. ZAC recruitment doesn't depend, its recruitment doesn't depend on that, but its activation does. That's the distinction.
Okay.
Okay?
Yeah.
So Rachel, I have one other question. What does the stress do? I mean, when you, especially when you have, I mean, do exactly, and you have NS, you have oxidative stress, and maybe you could have nutrient starvation. What's common between all the stresses? I mean,
My view is,
causing.
I'm not saying everything is about ribosome collisions, but everything you've mentioned, we think leads to collisions. So amino acid starvation definitely leads to collisions,
Yeah.
which we show in our paper. Oxidative damage, I showed you the proline. We think it's also true in mammalian cells. So is methyl MMS treatment, the damage. Maybe I'll just go through the next bit, and UV irradiation. I'm going to show you.
Okay,
But I
great.
think I think the goal as scientists, our goal is to make things the most parsimonious models we can, frugal models. And a frugal model is that collisions are at the center of many stress pathways, input into collisions. So that's what I'm going to tell you here, which is, okay, anisomycin, cyclophosphamide, whatever. These are drugs. This isn't really a very natural stress. But as I mentioned, Bruce Magan had shown that UV irradiation also activates the so-called ribotoxic stress. So we went on to essentially just validate what he had said, which is that just like anisomycin, which leads to ZAC activation and downstream EIF2 alpha phosphorylation and junk activation, we show that UV irradiation in the same way leads to ZAC activation in these downstream events. It depends on ZAC, everything is the same. So we confirmed what he had said, which is that UV irradiation looks an awful lot like ribotoxic stress. But the question is, how does UV light impact ribosomes? Actually, Bruce Magan's model was that there were damaged ribosomes that got stuck. I don't dispute that there are damaged ribosomes, but I don't think they're the cause. Well, the way we think about it is, you're all familiar with the fact that UV irradiation leads to DNA damage and pyrimidine dimers and double-stranded breaks and recruitment of and activation of kinases in the nucleus, a MAP3 kinase in the nucleus that leads downstream to this, P38 activation again. I can tell you the time course of this activation is on the order of hours usually, not minutes. And I can tell you that the ribotoxic stress response that I've been showing you, these are 5-minute experiments. We expose the cells for 5 minutes to UV stress and we get huge activation of P38. What you can imagine I'm leading to is if UV irradiation causes DNA damage, it could also cause RNA damage and pyrimidine dimers. And we have the tools to think about this and ask whether that's true. And if RNA were damaged, it would lead to collisions. So we did 3 experiments. You've seen all of them before, and that's why I took the time to introduce them. First, we looked at simple polysome profiling. These are undigested, DJ. We looked to see no, I apologize. These are digested. These are digested. So we have untreated cells and UV irradiated cells. You can see in untreated cells, there's a small number of disomes, but in the UV irradiated cells, there's more RNase resistant disomes and trisomes, consistent with the idea that ribosomes are colliding on damaged messages. We did a ribosome profiling, which is the sequencing experiment, to see what the problem might be. You remember we had 21 to 28 nucleotide ratios in untreated cells. You can see in UV treated cells, there's a huge change in the distribution of reads. And that distributional change is consistent with ribosomes being stuck here, which is exactly where you'd be stuck if the first time you encountered a message as it came into the A site was the damage enters the A site, the first problem is going to be decoding. So that's consistent with ribosomes getting stuck on damaged sites. And if we look at the distribution again of the 64 codons in untreated versus UV treated cells, these are the 21 mers, we can see that the places where ribosomes are disproportionately paused under UV irradiation are at codons that contain pyrimidine dimers. And this underestimates it because some of the pyrimidine dimers are between codons. So we thought all that was consistent with the model that in fact mRNA damage is the problem that leads to the ribotoxic stress response. But we did one final experiment, which was recommended by my colleague, Jeff Corden. He says, because our stress was in all this, okay, everybody's going to say, but how can you exclude that DNA damage is activating these pathways? Of course, DNA is getting damaged. So Jeff suggested that we take an RNA transcript in vitro, UV irradiate it in vitro, and then transfect it into cells, and to see whether simply putting in damaged RNA into cells was sufficient to activate these pathways. And it worked beautifully. So this is EGFP mRNA, either untreated or treated with UV light. When we put the UV irradiated sample into the cells, you get activation of ZAC, P38 junk, and the integrated stress response. And in fact, EGFP protein is no longer made because the RNA is damaged, whereas it is made in the untreated cells. So this was a beautiful demonstration that mRNA damage itself is sufficient to activate this whole pathway. We can come back to other questions at the end, or you can interrupt now, it's fine. This is just a summary of Collins' data. So Collins' data together suggested that at the heart, downstream of ribosome collisions, when collisions are overwhelming the mRNA quality control pathways, is the recruitment of this ZAC alpha, which is a MAP3 kinase, which is in fact important for the recruitment of this GCN complex, which leads to the activation of the integrated stress response, which blocks further initiation and gives cells the time to survive and recover. The integrated stress response is a way for the cells to get past the damage or the stress. And that failing that, what we think happens is that ZAC alpha becomes phosphorylated, probably falls off the ribosome to activate downstream MAP2Ks and MAP1Ks. And I just want to remind you here, because it's going to become important, the JUNK stands for June N-terminal kinase, which is upstream of June, which when phosphorylated is a transcription factor that goes to the nucleus to bring about downstream events. And that these are generally apoptotic events. And so the way we think about it is that there's some mechanism in the cell to sort of measure the number of collisions. When collisions are moderate, the survival pathways are activated. And when collisions are out of control, then the apoptotic pathways take over. So that's story one, and I'm going to tell you quickly another story, and then I'm going to try to connect them. So the next story is another postdoc in the lab, Niladri Sinha, who was interested in collisions and identifying factors involved in collisions. He shared a bay with Colin, and they were good friends. And so he did a similar experiment. He used emetine, which is a good drug for causing collisions, but the exact same experiment, no dose, medium dose, high dose. In this case, he decided to do a mass spec experiment with Wade Harper's lab in Albin Ordero, where they fractionated these emetine-treated samples across sucrose gradients. He isolated 11 fractions across the gradient, then TMT mass spec analysis of each fraction in triplicate for each sample to identify factors that are disproportionately enriched on deep ribosomes in the presence of drugs that cause collisions. So there's lots and lots of data here, and I'm going to just show you a quick slice. This is actually fraction 8, and he's doing a comparison of the orange traces to the blue traces to see what's retained on colliding ribosome samples. And what you can see, on the x-axis is enrichment, on the y-axis is significance. You can see there's a whole bunch of interesting genes, and there's lots to dig in here for people that are interested in this problem. We should be studying lots of them. But the one that Niladri chose to study was, of course, this factor here called EDF1, which stands for endothelial differentiation factor 1. It clearly was very enriched, and it was quite a significant hit. To convince you that this wasn't just true of fraction 8, I'll show you the quantitation across the whole polysome profile. You can see there's very little enrichment of EDF1 in the 40S, 60S, and 80S fractions, but that it's significantly enriched all the way across the deep gradient. I'll show you another of those figures that we were talking about before for ZAC. And you can beautifully see that in this case, unlike ZAC, EDF1 migrates in fraction 1 entirely for the untreated samples and in the high drug treated samples. But with low drug, it's completely shifted into the deep polysome fractions, consistent with the idea that it might be binding to collisions. So Niladri did a lot to characterize this factor. I'm just going to tell you a very few things about it. I'm going to tell you where it binds, which he was able to determine in collaboration with Roland Beckman's lab. And this is the answer. I could show you a lot of data on this, but I'm just going to show you the answer. So this is actually a cryo-EM structure of a trisome. This is actually a collection of yeast ribosomes. We also solved it on human ribosomes, but the view here is a little better. This is the lead ribosome. This is a colliding ribosome, and this is the next colliding ribosome. So it's a trisome structure. What you can see is that MBF1 is the yeast homologue of EDF1. And what you can see is that MBF1 is found here in the small subunit of the leading two ribosomes and not, sorry, in the lagging two ribosomes, but not in the leading ribosome. If you look carefully at where this factor is bound, it sort of wraps its way around the messenger RNA. And I can tell you that previous work from Beth Grayhack's lab said that this factor was critical for preventing frame shifting. So it's bound in a beautiful position. What I like best about looking at this structure is that this factor really isn't bound in the lead ribosome, which gives some understanding as to how this factor might only bind to colliding ribosomes and not to simply elongating ribosomes. The structure doesn't immediately reveal why it only binds to the lagging ribosome, but I can tell you that there were 23 amino acids missing. And we think those 23 amino acids point this way, if you can see my arrow, and maybe a bridge to the sort of leading ribosome, and might provide the specificity. And we have data to suggest that's true, which I'll show you in a few slides. So we think it's a beautiful hit, sort of consistent with, a role in recognizing colliding ribosomes. Naladri did a number of other experiments that you would do once you know you have an interesting factor. He deleted it, he overexpressed it, he asked what changed. I'll show you one of those experiments. He took wild type cells, HEC293 cells, or deletion lines with an EDF1 deletion. And he actually repeated this mass spec experiment to see if there were any factors whose binding to colliding ribosomes specifically depend on this factor EDF1. And he got a collection of genes, as one would. This is again a mass spec experiment. What we were looking for here, this is a ratio of ratios that we're looking at in delta EDF1 over wild type. We're looking for factors who depend on EDF1 for their recruitment. And there were fewer factors that emerged. I can tell you that GEGF2 and 4E2 emerged as very interesting hits. These are factors in the field that are known to be involved in translational repression. And Naladri was able to show that these factors were critical to shut down translation on messenger RNAs where collisions were occurring to prevent ongoing sort of translation of a bad message. And I'm not going to show you any data, but I'll come to it in the model and you'll understand it. So those are very believable hits and they were validated. What I want to focus on, however, is Naladri's final experiment in this study, where he was thinking about, okay, EDF1 stands for endothelial differentiation factor. It had previously been shown to be a co-transcriptional activator that actually works together with Jun in the nucleus to activate transcription. So to sort of get a handle on that, Naladri did a simple set of experiments where he took cells that were either untreated or treated with a low dose of emetine to cause collisions. And first he did a simple experiment just to ask what is the transcriptional output or the report on collisions? What happens to cells transcriptionally when collisions are induced genome-wide? And then he asked, how does it look different in the EDF1 knockout line in the untreated versus the treated cells? I'm just going to show you a tiny bit of those data. What's shown here is a volcano plot of emetine treated versus untreated cells at 120 minutes. And this is the sort of the transcriptional output. So you can see there's some number of genes who are upregulated transcriptionally with high significance. And if you were a transcription person, you would probably think that this looks a little bit like an AP1 transcriptional program. And consistent with that, at the top of it is that at the top of this is Jun, sort of in significance and fold change. So Jun seems to be, Jun activation, transcriptional activation seems to be downstream of the collision activation pathway. We went on to ask, are these factors or are these genes that are upregulated dependent on EDF1 for that upregulation? And I'll tell you that most of them are, and I'm just going to show you the data for Jun. This is now untreated, emetine treated at 30 and 120 minutes. And you can see what I just showed you, which is a substantial upregulation of Jun at 120 minutes. And that activation is significantly diminished in the delta EDF1 background, consistent with the idea that EDF1 is contributing to that transcriptional coactivation. So this, I'm going to try to clarify all of this, but this was sort of the model at the end of Naladri's paper, is that collisions occur. EDF1, this relatively small protein, is specifically recruited to the collision interface. That in part, EDF1 is responsible for recruiting these factors GGF2 and 4E2 that are known to provide an initiation block that in principle is a sort of survival pathway. It's helping the cell to not keep making proteins from these defective messages. And failing that, EDF1 activates this transcriptional program at the heart of which is June activation, which is known to be essentially an apoptotic program. So that's where Naladri's story ended. So what I want to do in the last little while is try to bring these together, because I've just told you two stories about two proteins and two postdocs who seem to have factors that do sort of related things. And I want to tell you how we're thinking about them together. What I told you from Colin's work is that ZAC is recruited to colliding ribosomes and is critical because it activates this signaling cascade downstream. And of particular importance is that JUNK gets activated. And as I mentioned, JUNK is a kinase that's upstream of June. And that when June gets phosphorylated, June goes into the nucleus to promote an AP1 transcriptional response. I also just showed you that there's this other factor, EDF1, that seems to be recruited to colliding ribosomes. And somehow downstream of collisions generally is an EDF1 dependent transcriptional program, and consistent, previous reports had said that these factors are both working together to have a transcriptional response. They didn't know that it was collisions that it was responding to, but they knew there was a response. So what I haven't told you yet is that EDF1 is present in great excess over June, first of all. And what we began to wonder is, given that EDF1 and June interact together in the nucleus on DNA to promote a transcriptional response, is it possible that these factors interact in the cytoplasm, and in this way that EDF1 controls the activation of June? And it would control the activation with June because under normal conditions, EDF1 is in great excess over June. June is bound to EDF1 and is blocked from being activated. And only when collisions are sufficiently abundant is EDF1 recruited away from June, thus allowing June to be liberated in order to be activated. And what this would do is it would provide a mechanism by which ZAC could be sort of activated all the time. Collisions occur, ZAC gets activated, JUNK gets activated, but nothing is consequential from that JUNK activation until enough June is released from EDF1 to be allowed to be phosphorylated to then move into the nucleus. And so EDF1 would essentially be the regulator of June activation. So that's the model that we sort of came up with when thinking about it. And we had looked at some data as well. And I'm just going to show you a few pieces of data to support this model. The first thing I'll tell you is we've switched to HEC-HAT cells. Everything else I've been telling you has been in sort of HEC293 cells. These are keratinocyte models that naturally are responsive to UV light, and they're just beautiful translating cells. They have huge polysomes, and all the pathways we're interested in are beautifully activated by UV light. So this is June activation, integrated stress response, and this is actually ubiquitination of the ribosome as a function of collisions with UV radiation. So we've begun to test different features of this model. And the first one I'll show you is this. The first prediction, and in some ways the most important prediction of this model, is that if EDF1 plays a role in negative regulation of June, you would predict that when you delete EDF1, June would be constitutively activated and even hyperactivated under conditions of UV irradiation. So we did a simple experiment with untreated or UV treated cells, and we either had a scrambled siRNA or a ZAC knockdown or an EDF1 knockdown. And we're simply looking at June activation here. And what you can see is that even in untreated cells and in EDF1 depletion background, there's some constitutive activation of EDF1 of June, which likely is responding to the occasional collision that Yi was asking about. You can also see that under UV irradiation, there's a greater background. With UV, this is sort of wild type background. And in an EDF1 deletion background, the signal is greatly increased. And that signal increase is dependent on ZAC, which it should be since we think ZAC is upstream of JUNK, which is critical for phosphorylating June. So that's a nice piece of data that's consistent with the model. We're trying to get an overexpression line set up, and we're hoping that that piece of data will yield the opposite result. The other prediction of this model that we've got some data to support is the idea that is to see whether these two factors associate in the cytoplasm under normal conditions, and whether in the presence of collisions, when EDF1 can be recruited away, they no longer associate with one another because June has been released. And this is an IP where we have a strep tag version of EDF1, and we're looking at its interaction with June. And you can see in untreated cells, EDF1 nicely associates with June, whereas in cells treated either with intermediate doses of drug to induce collisions or with UV light, that interaction is lost, consistent with the idea that the recruitment to ribosomes liberates June and EDF1 no longer binds to it. The final experiment I'll show you is here, and it's an important one. Another prediction of this model is that if this model is true, it should be true that a mutant of EDF1 that can't be recruited to the ribosome will block the activation of June because it will hold on to June because it doesn't get recruited to ribosomes. And so we've been looking for a mutant of EDF1 that doesn't bind ribosomes to the collisions. I mentioned when I showed you the cryo-EM structure, there were 23 amino acids at the end unaccounted for that we thought might be that critical to binding the collision. And we've shown that's true. And we use that mutant here to ask the question. So we have wild type cells or cells with the delta 23 mutant of EDF1. What you can see is the activation of June in wild type cells, but that activation is blocked in cells that have an EDF1 variant that can't be recruited to ribosomes, again, consistent with this model. So that's sort of what we're thinking about. We're thinking that this collision that, we think collisions are important. We think the cell has to assess how many collisions there are in order to determine how much of what part of the pathway to activate. How seriously should they take this insult? Should they just activate the integrated stress response, or should they instead activate this more serious apoptotic response? And we think a key part of this that's been unappreciated is this negative regulatory role of EDF1 in keeping June protected from activation until its collisions are sufficiently high that all the EDF1 is recruited to ribosomes, liberating June to turn on this pathway. So we're pretty excited about this model. I think it's kind of an exciting idea, and it helps us to sort of think about how general these pathways might be. And this gets at Yi's question. I think lots and lots of damage leads to collisions. I think starvation leads to collisions, oxidative damage, methylation, alkylation damage, lots of things lead to this sort of damage. Sort of in terms of where we're going, our lab's trying to think a little bit more broadly about these pathways in a variety of different types of cells. You can imagine that some cells, for example, neurons that are long-lived, may very well have upregulated the number of factors, the amount of factor present to deal with mRNA-specific events, whereas cells like pancreatic cells that are just cranking out proteins may have become immune to sort of the occasional collision because they need to be so translationally active. And so we're trying to look at the balance of these three sort of outcomes over in different cell types and different stresses and different collisions or conditions to understand how cells may sort of balance ribosome homeostasis with load and then stress response. And with that, I'm going to stop. Thank again the three main players in these stories. Colin Wu did the beautiful work in yeast and all the work on ZAC. Naladri Sinha is a postdoc in the lab who's done all the work on EDF1 and with June signaling. Jake Sabo is a new student who was very happy to work on the project. This is our recent post-COVID picture, which captured us all in a strange moment. Nobody looking at the camera. I want to thank my colleague Sergi Rougeau downstairs, who really helped us all along the way with the MAP kinase field, and his student Amy Peterson, who helped with making ZAC knockout lines. Alban Ordero, who's now at the Sloan Kettering, was a postdoc in Wade Harper's lab, who's just been instrumental for our mass spec data. And Roland Beckman's lab for collaborations on cryo-EM and his student Katrina Best. And thank you for listening.
Well, thank you, Rachel. This is a wonderful talk. So any questions from the audience? I think I will ask the first one. So the two-hit model you propose, is that specific for mammalian cells, or is it also true for yeast?
Yeah, that's well, Miguel, whoops, my student Miguel would be happy to hear that question. MBF1 is a homolog in yeast, yes. We spent a little bit of time in yeast seeing if there was a MAP3K that was equivalent to ZAC. And I'm not sure it exists there. So we failed to have data to support the idea that it does. There's some evidence that in certain yeasts, not the actinomycetes, but the basidiomycetes maybe, that there might be a ZAC homolog. We haven't found it. But things are a little bit different in yeast as well, which is that GCN4, which is sort of the June equivalent, right? In that it's downstream of the integrated stress response. It's activated by translational response, by translational control downstream of GCN2 activation. So we think it's probably going to be different there. So we think, some of the proteins are there, some are not there. And we think, the summary is that in yeast, I'm not sure this leg exists, the ZAC leg. I'm not willing to say it's not there, but
Okay.
I don't know that it exists.
Okay, DJ.
Hey, Rachel, that's a fantastic, very clear seminar. It's great to follow what's going on in your lab now. So I have a very kind of naive question about the generality of this model. So if you compare the conditions that are known to activate the integrative stress response, how many of those conditions that they do not activate your ribosome collision dependent, like say ZAC, for example? How many? Is there any do these two sets of conditions completely overlap, or there are some conditions that can only activate one but not the other?
Well, yeah. I mean, what I can tell you is I think. You tell a story when you publish a paper, right?
Yeah.
So I told this beautiful story, which is lovely, right? Which is, we think the first thing that happens with collisions is they activate the survival pathway, and failing that, they go on to activate this apoptotic pathway. The truth is, Colin tried hard to get conditions where just this was activated and not yet this. And we didn't find them. We're working hard at it now because I think we have better tools than we did then. I think it logically makes sense. I think the cell has to measure it, but so that's one answer. Is I think the collision aspect, which is central here, and we know that ZAC is important both for this integrated stress response and for this sort of canonical MAP kinase thing. The collisions are central to it, and ZAC is central to it. But maybe I should add, just to remind you, that this is just one version of the integrated stress response that's triggered by the GCN complex and is a ribosome thing, right? GCN is definitely a ribosome thing, but there's PERK and PKR and HRI and these other pathways, right, that activate the integrated stress response completely differently by proteotoxic stress, by double-stranded RNA, viral responses, and by cytoplasmic proteostasis with HRI. So I think the integrated stress response is much broader.
It's broader, okay.
It's much broader.
Right.
But I think there's super interesting questions to ask about how to me, I just can't imagine how the cell integrates this. How does the cell decide that this is what it wants now? There's got to be some measured response that we haven't been able to capture.
Mm-hmm.
Joshua?
Hi, can you hear me?
Yes, I can.
Great talk. I really enjoyed that, Rachel. Beautiful work.
Thank you.
I thought I saw on one of your slides that when you UV irradiated cells, in addition to seeing increased disomes, you saw there was a decrease in 60S subunits. Did I notice that correctly? And is that a separate response, or what's going on there? Is that something that's known, or?
Yeah, I'm trying to think what it is. Yeah.
Polysome slide, at least
I think it was this one. Yeah, it was this one.
Oh, yeah.
I'm trying to think because this is in the context of an RNase digestion. That's gone. Yeah, that could be interesting.
40S is unaffected, I guess.
Yeah, that's correct. Yeah. Yes, you're exactly correct. I don't know if that's known. It's definitely that I look at that and I think that looks real. Yes. What often, the reason I'm hesitating a little is normally if you're not looking at a digested sample, you get these half-mers, I'm sure you're familiar with them, you know, that those are usually the 40S that are kind of, you usually deplete 40s disproportionately. So yeah, this is weird. This is weird.
Okay. Well,
And
thanks
I don't
for,
know what it is.
Okay. Daisuke?
Hello, great talk. I really enjoyed it.
Thank
I
you.
have two questions. One is your transfection experiment, in which you transfected the UV radiated messenger RNA.
Mhm.
Why is it that like those bad messenger RNAs specifically eliminated, and instead it's activating systemic ZAC response? Was it just because you have way too many copies of messenger RNAs going into the cell?
Yeah, I mean, I think that's right. I mean, I think certainly, you know, the way I presented it, and I think that's probably generally true. I think the first thing a cell's going to do is try to fix the problem. And the first thing to do to fix the problem is to get rid of it. Did we see, you know, certainly our goal there, Colin doesn't tell me when things don't work. He just tells me when they do work. I suspect that it took a few experiments to get that working, and he put in a good amount.
I see.
So
Okay.
the goal there was, you know, essentially the goal there was to titrate into a cell, specifically a collision.
I see. But then there are collision specific QC pathways.
Yeah. I mean, that stuff all exists, and the rest of my lab is usually studying
I
that.
see.
And that's a beautiful pathway.
I see. And the second quick question is, what do you think is a contribution of this collision-based UV-dependent cell death pathway compared to the DNA based death? Like, for example, if you remove ZAC, would they be more susceptible to UV damage or more resistant?
Well, that's a great question. I know almost nothing about DNA damage. But I can tell you in our paper, we had a ZAC knockout, and it is more resistant to apoptosis. So, you know, at least in a first pass on a first, some time course that we did, that's its phenotype. And that kind of actually is why it came out of the CRISPR screen, right? We looked at survival on anisomycin. So anisomycin causes collisions, which leads to apoptosis. We also got all the apoptosis genes. We got all the, I guess, the BCLs or whatever they are. That whole pathway lit up. And ZAC lit up there because it made the cells more resistant to apoptosis.
Oh, that's great.
Long term, I recognize that the DNA pathways are critical. So I don't know how to balance those. I'm sure there's interesting things to be done.
Thank you.
Okay, Michael, you have the final question.
Hi, Rachel. Hey, really beautiful talk. And I apologize if I missed this, but do you have any insights into how ribosome collisions actually lead to ZAC activation and ZAC autophosphorylation?
We're working on that. We do have, what I can tell you that I didn't share with you is we have a domain, and ZAC is, if you look at it now in AlphaFold, you get scared and think you shouldn't work on it. You would probably get excited and think I really should work on that. But there's a little domain at the end that's been implicated in binding, and also by a guy named Simon Becker-Jensen. Sergi had told me that my colleague that MAP3Ks usually cross-phosphorylate one another, and we did a nice experiment in the paper where we have the endogenous gene and a transgene with a tag on it so we can distinguish. And it turns out that experiment very clearly showed that it was autophosphorylating. It was a cis reaction. So we're thinking it's a monomer. We're working very hard on getting binding mutants and kinase dead mutants. We've been working hard with Roland Beckman on getting a structure. As with many things in the I don't know how well you know how the cryo-EM thing happens in ribosomes, but the best experiment is you tag your protein, you pull, you get ribosomes, you solve the structure. And when you do that with ZAC, the only thing that's on a grid is disomes. So I think we're right. I think it binds disomes, and you can solve the disomes and you can't see any ZAC. So, which is a pretty common output.
Okay.
But, I guess I imagine that ZAC is tagging along with ribosomes, and when the collision comes along, domains get rearranged and result in this autophosphorylation. So we need to work harder at that mechanism.
Great, thanks.
Okay, thanks everybody for coming, and thank Rachel for giving a wonderful talk. And well, see you guys later.
Thank you very much, Chief.
So Rachel, we will have to